Many believe Australia has lost a crucial opportunity to curb carp by shelving a brilliant scientific project being conducted at the CSIRO. It's called daughterless carp, and as it suggests, it's technology that induces carp to produce only male offspring.
So that basically they can't keep breeding in the long term, that the population just becomes swamped by males, and eventually, over a period of time, they'll die out.
Dr Ron Thresher and his team began their work in 2000, taking their cue from entomologists who had bred all male populations of insects, such as fruit fly, thereby rendering them infertile. The team decoded the gene technology that can produce the same effect in fish. The evidence, what's now left of it, is seen here in a secure marine laboratory at the CSIRO in Hobart. These zebrafish are related to carp, but because they have a much shorter breeding cycle, only about 2 months, they're really the freshwater equivalent of laboratory rats. But these fish are also much more. These are the 4th generation spawned using this amazing technology.
You look at them, they look like perfectly normal zebrafish. We've done lots and lots of experiments looking at fecundity, checking the eggs and everything. The males that are carrying the construct are perfectly normal. They grow the same size as a normal male, they're competitive with normal males, they're just as fertile as normal males, but they're all males. And that's the trick.
From July 1st this year, funding for the daughterless carp project stopped, unofficially, because the technology was still several years away from adoption, and officially because there was simply not sufficient funds to support the entire range of carp control measures.
To an extent, we've proven what we wanted to prove, which is that the technology works. You know, doing it in zebrafish, the next critical step is finding out whether it works in carp. And if there's a sad bit, it's because we're halfway through that experiment and we're going to have to terminate it before we actually get the definitive answer.
Dr Thresher says that answer would have been known within 2 years. To start from scratch now would take 5 or 6 years.
The cost of finding out really is, you know, a few $100,000 compared to the $4 million we've already invested in this exercise. It seems like sort of a shame to stop it at this point.
At Auburn University in Alabama, USA, European carp implanted with the CSIRO's daughterless gene technology are also on death row. About $60,000 is needed to keep those fish alive and that experiment running. Dr Ken McColl believes the Koi herpes virus, combined with the daughterless technology, would prove a lethal combination. In mothballing the daughterless project, he says we've lost a crucial opportunity.
All of the modelling shows that KHV will work very well, but if we can get the two technologies going together, then everything indicates that we actually have not only the chance to control carp, but actually to eradicate carp. And within a reasonable time frame of 20 to 30 years. So I think it's a major loss to the nation, really, if we can't do something about maintaining both aspects.
It's the combination which is deadly to these things, and that's the point we're trying to get across to everybody, that, you know, picking and choosing a winner is probably not the best way to do it. You want to have these things working in synthesis, and I think that's where we really need to go.
The potential of the daughterless gene technology extends far beyond the pesky carp.
The technology we're developing is generic, not genetic, generic. We're looking for gene constructs that can be readily adapted to a whole range of pest species. So the genes that we're targeting in zebrafish or carp are much the same as the genes you'd find in a cane toad or tilapia or a whole range of other lower vertebrate pests. And in fact, the basic technology would even be applicable to mammals, although with mammals it's much more complicated from a population control perspective, because their physiology and their ecology is much more complicated than lower vertebrates. But certainly this technology could be readily adapted to something like a cane toad. Probably you'd have a working construct within 6 months to a year.
The daughterless carp project was funded by the Murray-Darling Basin Authority, which in turn is funded by the federal government. During the past 7 years, the authority invested $6 million in the native fish recovery program, aimed at restoring native fish numbers in the Murray-Darling Basin. Till recently, the daughterless technology was regarded as a key hope for future carp control. But its progress was deemed to be too slow, and there were doubts about its effectiveness. In a statement, the authority says,
Whilst we believe the daughterless technology for carp has merit, it's a potentially long-term and expensive investment with no guarantee of success.
so after an independent scientific review, funding was stopped. The authority says it will
continue to support other control options, measures such as the research required to enable the registration and release of cyprinid herpes virus, which is likely to take a number of years to complete.
KHV is a species-specific technology. It'll only work for carp, and that's what we're relying on. Daughterless, on the other hand, is what we call a platform technology. It can be adapted to any species or any species of fish that has that same sort of reproductive strategy. So, yeah, it has international significance as well as local significance.
That must be rather heartbreaking to see it shelved for lack of understanding or political will.
Yeah, it is. I would love to see both of them go ahead because it does offer a real opportunity to make a difference.
The daughterless technology has gained widespread interest from overseas. After all, it combines three prongs of attack.
One that is lethal to female, one that sterilizes female, and we have a third construct in test in carp in the United States, which sex changes fish. And these are all different ways of achieving this 100% male outcome.